Hello everybody, my name is Roman Bauer and today I'm going to talk about agent-based modeling and simulation for biological and medical research. But first of all, I would like to thank the organizers for having me. So I'm interested in the changes that occur during the human lifespan. And as I've shown here, there are numerous very critical and distinguished time periods during the human lifespan. These are, of course, just a small sample of those. But for instance, the gastrulation period or the event of gastrulation is arguably one of the most important events that occur. And actually, according to developmental biologist Louis Wolpert, this is actually more important than birth, marriage, and death. And then there is, for instance, neurulation, which of course is essential for the developing brain to occur. Then the birth, which also has implications, of course, with regards to how the brain structure and function organizes itself, and childhood, adolescence, and adulthood, during which all our brains are changing continuously, and then finally death, which is preceded usually with degeneration. And so from a medical perspective, one can also associate the lifespan with very specific medical conditions, for instance, anencephaly before birth, or autism spectrum disorders, and Alzheimer's disease. And so what is important to notice is that the ever-changing brain, for instance, in this case, raises a lot of scientific questions. And as a computational modeler, if I want to study those systems, then one of course needs to identify the most crucial components and simplify the computation model such that one can address specific scientific questions. And these of course differ for different labs, for different institutions, and so it is important to, before even starting to do research, to investigate what are the computational approaches that are available and which ones are most suited for a given research question. And additionally, what is important is that the computational modeling approach is something that is continuously being refined. So the initial situation is that one has some kind of experimental data for a given system, and then one uses this experimental data in order to produce or generate a computational model that of course makes certain simplifying assumptions. And then one simulates this model, one produces certain other results, certain types of outputs, and then one can compare with previously existing results from the literature, which usually will be experimental results or certain pre-existing knowledge. And depending on whether these results match, then one can also make, of course, certain predictions, which is, I think, one of the most crucial, let's say, outputs of a computational model, in my view, is not only to be able to model what is already there, what is already known, but also to say, okay, given this simplifying assumption and disagreement with existing experimental data, we would expect to see X and Y and so on. And so then this brings in the experimental stage again. So a computational modeling can never be understood as an endeavor that is in isolation of the real world and experimental evidence. So these predictions can then be tested experimentally, and if those match, then that's great. Then we can add further data and make or render the computational model essentially more powerful, increase its explanatory power. If not, then of course one needs to go back and see what could have been the reasons. And so there is this continuous refinement of the computational modeling, and this is I would say in most cases a never-ending task, because there is always some kind of data, especially in a biomedical context, that one can include on so many different spatial levels as well as temporal levels. And one of the computational approaches that I've used in the past, that we're using in my lab, is so-called agent-based modeling. And there the computational modeler has access to certain biological fundamental behaviors. So these are biological primitives based on which the computation modeler can create more complex computational models. So this can be understood similar to Lego, for instance. Those Lego toys, when you can create maybe some car or other more complex toy, you build it using elemental building blocks. And this is quite similar to agent-based modeling, because in agent-based modeling, usually you have access to those biological fundamental behaviors, which could be, for example, the behavior of changing a physical property. So, for example, a cell body could increase its size or shrink its size, or change some other aspects of its physical properties. Or it could move, so it could change its location in three-dimensional space or two-dimensional space. And this could be, for example, cellular migration, which is, of course, a very crucial type of behavior during development, including the development of the brain. And then there is also the proliferation, for example. So cells can divide and give rise to other cells using cell division, or also a cell body can, for example, create an initial segment of a neurite. So let's say an axon grows out from a cell body, and this can also be understood as the spawning of a new type of element. So you understand that these are very generic terms, but they can be adapted to the specific context. Then there is the secretion of substances. So cells secrete in certain substances into the extracellular matrix, for instance, and then those can diffuse, and they can affect other cells which detect them. So cells don't only secrete, but they also detect. It's like writing information into the world and reading the information. And then there is, of course, also dying, and for instance, apoptosis, which is a very active process, actually. And cells can not only give birth to other cells, but they can also die. And in many cases, this happens during brain development, for example. About more than half of all cells just die naturally during normal development. So it's a very common phenomenon. And then there is also the bonding behavior, where cells can, for example, mechanically bond to one another, or there's some kind of mechanical link created between cells. So this is also one of the biological fundamentals which the computational modeler usually has access to when using agent-based modeling. And so this is just to kind of give an overview of the context as a computational modeler in agent-based modeling, if one wants to simulate a complex biological systems, such as, for instance, the brain. But of course, this could also be applied to entirely different systems, like a liver or a kidney, and so on. It doesn't even need to be biological. Also, this is also something to emphasize. But in this context, in this talk, I'm only addressing biological and medical aspects. So one example that I would like to showcase here, just so that you maybe can better understand or get a better idea of what this means in practice as a computational modeler, we have looked at the so-called superficial patch system, which is a patchy system of connections between neurons in the superficial layers of the mammalian cortex. So this has been observed in all mammalian cortices, except it seems in rodents. And here you see an image where here in the middle is a black, in black is the injection site of a tracer, and this tracer labeled those patches. So you see those regions which are somehow not uniformly labeled, but you see those patches. And so for some reason, this site in the middle is connected in a patchy manner to its surrounding cells. And one question that arises is not only, of course, what is the function of this? This is a very interesting question, and people have been looking into this. But one question that is also arising is, how does this develop in a biologically realistic manner? Because it's still not really understood how this develops. And it turns out that actually Alan Turing proposed a mechanism that can explain this. And here you see me sitting next to Alan Turing in a park in Manchester, I think this was. And Alan Turing published a paper in 1952 called The Chemical Basis of Morphogenesis. And in this paper, he explains that actually, and he shows and demonstrates that substances that interact with one another can actually produce patterns, including patchy patterns, but also other types of patterns, like, for example, the stripes on the coating of a zebra, for example, can also be explained with such systems, or Turing systems, or fingerprints, for instance. There are a wide range of different kinds of patterns in nature, and many of those could develop based on this system that Alan Turing proposed. And one subsequent model that is actually based on Alan Turing's seminal work is the so-called Girer-Meinhard activator inhibitor model. So in the Girer-Meinhard model, we have two substances, an activator substance and an inhibitor substance. And the activator substance is self-enhancing. So cells produce more of it, the more there is already there around the cell body, for instance. And the inhibitor substance also gets increased if there is a lot of activator substance. However, the inhibitor substance then decreases the production of the activator substance. So we have these two antagonistic kind of substances. And it turns out, if one then also adds diffusion, and then one has the right parameters for the production and the diffusion and decay and so on, then one can create actually patchy patterns in addition to other types of patterns. And here you see a simulation where we have a layer of cells. So this is a two-dimensional system in this case, where you can see here in those colors patches that actually developed from a Girer-Meinhard system. So each of those cells was secreting those substances accordingly. And after the patches have developed, we then simulated how the axons would be guided towards those patches and create arborization patterns, similar to what I just showed you before in this image. And here you see now this in a video, just to give a better sense of how this plays out in time. So here we started from a single cell that then first produces a plate of cells, again a two-dimensional system, and then we have several activator inhibitor models, so several Girer-Meinhard systems, which each produce a patchy system. And in this case, these different cells then are drawn, or they project their axons to the like in a homotypic fashion to the same type of cells, and they create those patches. And so this is essentially one potential, it's not proven yet, but it's a potential model for how the superficial patch system could develop. Now, there is also evidence, in addition to just the structure, we can also measure actually in nature what is the spacing between patches and what is the width between of the patches. And it turns out that if one does that for different species and different areas, that there is roughly a linear relationship with a slope of about 1.65. So the spacing is is about 1.65 times the patch width in a linear manner. And it turns out that just by changing one single parameter in the computational model and then simulating different patch systems, we actually get the same kind of, more or less the same kind of relationship. It's the slope is very similar, and so this is, I think, very strong evidence that this computation model could indeed be correct, or at least capture the most essential parts of the generative mechanism for this superficial patch system. And it's also, as you may have noticed, quite aesthetically pleasant, and we were published a few years ago, and it was also shown on the cover. So another example that I just want to show you, where we have applied agent-based modeling and simulated it, is the so-called cortical layer architecture. Again, this is part of the brain, and not only the mammalian brain, actually many other brains also have a layered cortical architecture. And what this is that different cell types are arranged in a layered manner, and we see this in lots of different species. But what is also interesting is that the dimensions and the cell numbers can differ a lot. So you can see here even orders of magnitude of differences between different species and even areas of the same species. And so one question that arises is, again, how can this develop in a biologically realistic manner? Can we use computational modeling to maybe gain some insights into this? And how can we also explain the variation across different brain areas and across different species? And so what we did in a few years ago is we created an agent-based model where we have a gene regulatory network that has this kind of state machine or state kind of dynamics, where we start from an initial proliferative state called S1, and then we can differentiate, or the cell differentiates probabilistically into different kinds of cell types, and there is also apoptosis, so cell death. And actually it turns out that cell death is crucial. So without cell death, we were not able to really produce this variety of different kinds of cell architecture or layer architectures. And so we have here an image where we see throughout time how the layer architecture develops in simulation, and you can see here that actually we can get such a clearly layered structure. And what is also important to note is that here in this case, all the cells interact mechanically with one another if they're nearby, so they really push each other. I have also created a video that you can access via this publication, which is available as open access, and so this shows how, in a biologically realistic manner, using agent-based modeling, we can demonstrate a potentially realistic model for how the layered architecture develops in the cortex. And this is also relevant from a medical perspective. So we actually then looked at different parameterizations of this model, and we could, for example, generate a much thinner cortex just by changing one single parameter. And this could, for instance, have implications with regards to polymicrogyria, where the cortex is much thinner as compared to a controlled, normal, healthy subject's cortex. You see here, this is much thinner and has many more folding patterns. And so this could be a way to better study how does polymicrogyria develop, and what could be maybe individual genes even, how could they be differently expressed, and how does this play out mechanistically. Similarly, we also found certain patterns reminiscent of what people have observed in autism spectrum disorders, where the cell body locations seem to be a bit less regularly arranged as in the control scenario. And also here, by changing apoptosis, we could actually gain similar locations of cell bodies that are a bit less regular than otherwise. Now, these examples that I showed you usually were simulations that were rather small, so they had maybe a few thousands of neurons or cell bodies. However, if you want to look at more clinically relevant systems, we need to, of course, scale up those simulations. And so what we did is we created computer code that is very, very efficient. And here you see one publication where we show how one can write efficient agent-based simulation code. And we created a software called BiDynamo, where a consortium of different institutions, so here you see the institutions of the BiDynamo consortium, where we have created a highly performing agent-based modeling software, and we can simulate very large systems with billions of cell bodies, for instance, or otherwise agents interacting with one another. And we published this rather recently, and we also provide this software as open source. And we have also a website with a lot of documentation where the interested audience member could have a look with example demonstrations. So we have demos that you can even run from your browser. You don't need to install BiDynamo necessarily. And we have a community of researchers that regularly meet. So if you are interested in potentially using agent-based modeling for your own biological research, feel free to get in touch with me or with the BiDynamo consortium in general. We are also very interested in didactic endeavors, so in training opportunities. We regularly participate in summer schools. So a few years ago, for example, we co-organized the Modern Code Developer Challenge that attracted thousands of students from all around the world. And this was, I think, a very, very nice event, because it also helped students to gain a better understanding of what agent-based modeling is and how it is implemented. So in this way, I would like to conclude by thanking my collaborators. I could not list all of them here, but I'm very thankful for the many collaborations that I had in my research. And I would like to thank the Combine Lab, my lab at the University of Surrey, which is a very lively bunch of people. And I would like to thank you for your attention. Thank you.